blue, not red. The blood of the horseshoe crab, Limulus polyphemus, is the most extraordinary biological fluid in medicine. It contains amebocytes.
Delaware Bay, eastern United States. Every spring, at the full moon and new moon high tides of May and June, the largest concentration.
The collection team has been working since 3 a.m. The optimal window is the 4 hours around high tide, when horseshoe crabs are most active in the shallows.
An adult female horseshoe crab, 45 centimeters wide, 4.5 kilograms, approximately 20 years old.
Horseshoe crabs are not true crabs, and they are more closely related to spiders and scorpions, part of the subphylum Chelicerata. The horseshoe crab's compound eye contributed directly to our understanding of human vision. Nobel Prize-winning research in Netherlands. The biomedical facility processes horseshoe crabs in partnership with commercial fishermen under a strict regulatory framework. Horseshoe crabs must be held in cold, well-oxygenated seawater for a minimum of 2 hours before blood extraction, allowing them to recover.
The extraction laboratory operates on a timed schedule. Each horseshoe crab spends a maximum of 30 minutes in the extraction frame. The extraction frame angle, 30 degrees from horizontal, is not arbitrary. At this angle, the horseshoe crab's the extraction needle enters at the arthrodial membrane, the only non-hardened tissue on the horseshoe crab's underside. Below this
cerulean blue. The color comes from hemocyanin, the horseshoe crab's oxygen-carrying molecule, which uses copper atoms instead of the iron atoms in human hemoglobin. What you are seeing in this tube is not just blue blood. You are seeing the reason that every injectable drug, every IV solution, every vaccine, 6 simultaneous extractions. Each animal will contribute approximately 30% of her total blood volume, the maximum safe extraction limit established by 30 years of research. 60 milliliters of blue blood. At pharmaceutical processing yields, this sample will produce approximately 15 milliliters of
Recovery begins immediately upon return to water. The horseshoe crab's physiology prioritizes blood volume restoration after extraction.
The facility biologist monitors every recovery animal individually. Her role exists because the industry learned.
Centrifugation separates the amebocytes from the carrier hemolymph, a process taking 10 minutes. 10 minutes.
Lysis, the breaking of the amebocyte cell membrane, releases the enzyme cascade responsible for the LAL test's extraordinary sensitivity. The final LAL reagent is clear, pale yellow, and completely unlike the vivid blue blood it came from. The return. Every animal collected this morning goes back to Delaware Bay this afternoon. The facility's license requires GPS documentation of the return location. She walks back into the bay as if the last 12 hours did not happen. The needle entry site at her arthrodial membrane has already sealed.
500 animals collected, 500 animals returned. In between, approximately 40 liters of blue blood extracted, processed into LAL reagent. At peak spawning, an estimated 1 million horseshoe crabs emerge onto Delaware Bay beaches over 10 days.
The bookgills are both the horseshoe crab's respiratory organs and the most visible indicator of its physiological state during handling. Health conditions. Recombinant factor C, or RFC, is a fully synthetic alternative to LAL that uses lab-grown enzyme.
The science says RFC is equivalent. The regulatory system says, prove it in every application, separately, with full validation packages.
Delaware Bay's horseshoe crab population has declined approximately 75% since the 1990s. From an estimated 1.2 million spawning animals.
Tagged individual number DLB2847, female, tagged at this beach 4 years ago at an estimated age of 16 years, recited 7 times.
The horseshoe crab's eggs are the primary food source for migrating shorebirds on the Atlantic flyway, particularly the red knot. This red knot needs 70 grams of weight gain in 2 weeks. Horseshoe crab eggs contain 61% lipid.
I know more about horseshoe crabs than most people alive. I know how many there were 20 years ago and how many there are now.
He handles every animal himself on the return journey. He says he does not know why he started doing this. It is not required. The crew does it efficiently.
The target is 7%. The result is 8.3%. The difference? Approximately 65 animals per year above target mortalities.
In winter, Delaware Bay holds its horseshoe crabs in depths of 10 to 40 meters, the animals virtually in sorts. The facility prepares for the season the way any serious harvesting operation prepares, with attention to every variable that affects the out. She will run 240 LAL tests today. Every one of them depends on blue blood from an animal on the other side of the country. The IV infusion she prepared will enter a patient's bloodstream within the hour. That patient, whoever they are, wherever they are, will never know.
Delaware Bay, 450 million years of horseshoe crabs, 50 years of pharmaceutical extraction. The bay is patient with both of these timescales.
She does not know she is ancient. She does not know she is rare. She does not know that her blue blood is in a freezer in New Jersey.
The tagging program has been running for 11 years. Of the facility's tagged animals, 23% have been recaptured in subsequent years. Scanning the tag now. Confirmed. The database shows she was last processed 3 years ago. 8 to 5 milliliters collected, good condition. The Citizen Science Monitoring Program has collected spawning data from this beach for 19 years. A data set longer and more consistent.
A flipped horseshoe crab on the beach will die of desiccation within hours if not righted.
The facility holds these 12 animals as a wildlife service, not a commercial transaction. It happens several times per year.
Pharmaceutical execution. The pharmaceutical executives need to understand their supply chain vulnerability. Depends on wild animal availability in a specific
4,000 eggs per nest. A single female horseshoe crab lays between 80,000 and 100,000 eggs per spawning season, distributed across multiple nests. These juveniles will not be sexually mature until they are 9 to 12 years old. They will not appear in spawning surveys.
You have almost certainly benefited from this animal's blood. If you have ever received a vaccine, an IV, or a surgically implanted device,
Blue blood, Blue Bay. The same blue in 2 different states, one inside the animal, doing what 450 million years of evolution built it to do.
25 male.
Check. Total count is stable.
She is 6 years old, based on the tagging record. Her carapace width has grown from 32 centimeters to 41 centimeters, consistent with...
The director makes one note in the margin. Best season on record for welfare metrics. Population survey data concerning. Both things are true.
It's collection events 12. Animals processed 4,800. Return mortality 8.1%, 389.
The May full moon. It pulls the tide, and the tide pulls the horseshoe crabs, and the horseshoe crabs pull the shorebirds from South America. 450 million years, every May, the same beach, the same moon, the same ancient animals. The pharmaceutical industry found them 50 years ago.
the recovery stalled. The restrictions on bait harvest worked. The population was increasing. Then water temperatures in Delaware Bay.
The meeting table holds 4 competing legitimate interests. The pharmaceutical industry that needs the blood, the fishing industry that needs the bait.
This is where the data and the politics and the biology intersect for this year. This is how a harvest survives.
How old might it be?
18 to 20 years.
18 to 20 years.
Wide shot of the Delaware Bay at low tide in late summer. The horseshoe crabs gone to deeper water, the spawning beach empty, the red knots departed for the Arctic.
1 centimeter wide. 10 days old. The same body plan as an animal that outlived every mass extinction in Earth's history.
The biomedical facilities run on the tide schedule, not the work schedule. Horseshoe crabs are most accessible at high tide, in the 2 hours before and after peak.
Quiescent amebocytes. No degranulation, no gel formation. The sample is clean, endotoxin negative, safe for patient use. This batch will become 2 million doses of influenza vaccine. Every vial tested with LAL reagent from horseshoe crab amebocytes before release.
Forget the automated process. We are adapting the protocol. Use the benchtop centrifuges and keep them cool. The director has a hierarchy of concerns that she has never written down, but that governs every decision she makes.
The most important thing we ever did was decide that the animals mattered more than the quota. Everything else followed from that.
This moment, high tide, full moon, May, is the oldest appointment in this story. The horseshoe crabs have kept it for 450 million years.
2 blues, 1 ancient, 1 daily.
The first animal return sets the standard for everything that follows.
the crew member who returns the last animal of the day with the
visit. The marsh is part of the ecosystem. The facility's return protocols include returning animals to locations that maximize the...
Everything's stable. The crabs are well.
The acoustic telemetry study is the most detailed post-extraction recovery research ever conducted. The implant...
If the data says it helps the animals, we change the protocol.
Wide shot of Delaware Bay in winter. Ice forming at the bay's edges in a cold December. Conditions. Limulus polyphemus, the Atlantic horseshoe crab. It survived the Ordovician extinction, the Devonian extinction,
This brings us to the crucial question of what the horseshoe crab needs from us. As the data suggests, stronger regulatory protection is essential. We know how to do all three of these things. We have not yet decided to. We know how to do all three. The synthetic alternatives exist and are validated.
She has been doing this work for 9 years. She believes the work is worth doing, but it needs to end before the population cannot sustain it.
Guilt is for people who haven't thought carefully about the question. I have thought carefully about it. I know the necessity. I know the harm.
Fossilized horseshoe crab tracks from the Jurassic period are indistinguishable from the tracks on this beach this morning. The Delaware Bay, the ancient and the modern, the ecological and the pharmaceutical, the animal and the medicine, all of it in one bay, under one sky.
All right, everyone, that's a wrap.
Finally, my cheeks were starting to hurt.
See you all at the crab dock.
All right, team, look right at the camera. Big smiles, everyone.
The engine is running. Are we clear to cast off?
Ready when you are, Captain.
Lines away.
She has written the same 4 recommendations for 7 years. Year by year, they are being acted on, one completed, then another, then another.
In some of them, humans have found what they need, and the question of how they take it is still being answered.
This is the future that the data points toward times, if the decisions are made correctly and made soon enough.
450 million years. Every mass extinction survived. Every sea level change navigated. Every...